Okay, so today's lecture, we are going to continue from our last lecture. So basically, during our last lecture, we uh, we have looked at the process of passive transports. Okay, so passive transport, as we know, it involves uh, diffusion. Okay, where molecule diffuses from a region of that solute being concentrated, region of the solute being less concentrated. So the movement from high concentration to low concentration. Okay, down concentration gradient. That is passive transport. Yeah, so for diffusion, right? So, and then under passive transport also is osmosis. Okay, and then after osmosis, we are going to look at on facilitated diffusion. So basically, all these three: diffusion, osmosis, and also facilitated diffusion is the movement of molecules from high concentration to low concentration until equilibrium is reached. So until the process completes. Macam itulah, okay. So osmosis. So we are going to look at first on the osmosis. We've covered on the diffusion part. So now we are going to look at on osmosis. So let's look at what is osmosis. Okay, so it is the diffusion of water. So osmosis, it must involves water molecule. Okay, the movement of water, diffusion of water, the diffusion of water across a selectively permeable membrane from the region of lower solute concentration to a region of higher solute concentration until the solute concentration on both sides of the membrane is equal. Until the solute concentration on both sides of uh, of of the solution is equal. Okay, so if you look at in this diagram here, so the one that you have to take note, okay, diffusion of water across selectively permeable membrane. This one, from a region of lower solute concentration to a region of higher solute concentration. If you look at this animation on the side here, you have there's the beaker that contains two different solution. Okay, two different solution. So kita bagilah region A and also region B. So if you look at the two solution which is separated by semi-permeable membrane here, it only allows for the movement of water molecule. Okay, so if you look at the region that contains solution B here, solution B will have solutes. Okay, solutes. So this is the region that has a high solute concentration, concentration of solute, okay, is high. Okay, on this side here there is no solute, so solute concentration is lower solute concentration. So in order for the two solution to reach the same concentration, okay, the one that will move is water, kan? So this is water molecule will move across the membrane through the pores. Okay, a semi-permeable membrane is through the pores, right? So this is the process of osmosis. It only involves the movement of water molecule from lower solute concentration. So this region solute concentration is low, from low to high. The only molecule that moves is the water, kan? Water molecules. Okay. Until what? Until both side has equal concentration of solutes, kan? Equal solute concentration, or the both side has same concentration, same concentration of solution. Okay. Okay, so if you look at this diagram here, there's the tube, we call it as the U-shaped glass tube. Okay, so U-shaped glass tube. So a U-shaped glass tube with a selectively permeable membrane here, okay, separates two sugar solutions. Okay, so you have two solution that has different concentration of sugars. So kita bagilah side dekat sini, kan. So this is side A, so this is also side B. The other one is side B. Okay, so side A has lower lower concentration of sugar. The other side, which is the B side, it has higher concentration of solutes or the sugar. Okay, sugar. So Apa rules dia tadi? Water will move from lower solute concentration to the region that has higher solute concentration. So it says here a U-shaped glass tube with a selectively permeable membrane separating two sugar solutions. So so it is separated by membranes that have pores, selectively permeable. Selectively permeable, why? Because it only allows water to move. It doesn't allow sugar to move. Okay, so because only water can move, water will move until both solutions in region A and also region B has the same concentration. So as you can see, because solution in side B here, where it has higher solute concentration, in order to balance the solution concentration, so only water will move from region A to region B. So as you can see at the end of the experiment, because only water moves, region B, the level of the solution will increase because water from A moves to B, from low solute concentration to high solute concentration. That is the movement of water. Okay. So water moves, it says here, water moves from an area of higher, Casty, you have to take note, from higher to lower free. This one, it uses another term, higher to lower free water concentration. This region here, it has higher water concentration. This side here, it has lower water concentration. So water moves from where it is more to the region where it is less. Okay. So another factor, as you can see from this diagram here, okay, region A tadi and then this is region B. So region B has lower solute concentration. Solute concentration is low on this side. Okay, this side B side, solute concentration is high. When there's more solutes, when there's more solute at region B ni tadi, it requires more water molecule to be involved in the formation of what? Hydration shell. Okay, hydration shell. Kita dah belajar kan hydration shell, kan? So more water will be needed or required to surround the solute, causing the solute to dissolve in the solution. Okay, so when there's more water involved in the formation of hydration shell, causing solute to dissolve, kan? So therefore, side B will have less less free water available, less free water. Why does it uses the word free? Because water that does not involve in the formation of hydration shell, that causes the solute to dissolve. That's why it uses the term free. Faham? Okay, free water that is free, that is available, that can move. Okay, it doesn't involve in the formation of hydration shell. Okay, so on this side here, because there's less solutes, there will be more water molecule, and there will be more water molecule. When there's more water molecule will move from a region of higher free water molecule to a region of lower free water molecule. So vice versa lah, macam mana nak explainkan dia. You can either use water moves, saya repeat water moves from an area of lower solute concentration to a region of higher solute concentration, ataupun you can say water moves from an area of higher free water molecule or free water concentration to region of lower free water concentration. So you can use both. Faham? Boleh? Faham tak part ni? Boleh faham?
Boleh.
Boleh,
Okay.
madam.
Alright. So you have to look at the process really carefully. So at the end of the process of osmosis, what do you get? You get two solution that have the same concentration. Two solution that have the same concentration. Okay, so this is what we call it as equilibrium. So when equilibrium is reached, will there be any more movement of water? Kalau equilibrium dah hasil, dah reach, kejap je kelas. Okay, so that is process osmosis. Okay, so soalan saya lah tadi apa? Will there any movement of water if equilibrium is reached?
No?
Ada. Ya, ada. Walaupun dah reach equilibrium, both side has the same concentration, kan, concentration of solutes or solution. When equilibrium is reached, there will be still movement of the water molecule, but at the same rate on both sides. Okay, so istilah yang kita guna, no net movement, no apa zero net movement, kan? No net movement of water, tapi but actually there's, maksudnya apa? Maksudnya equal rate of equal rate of movement of water from both sides. Okay, good try again. Right. Okay. So this is another animation, as you can see. So from this animation, you can see that okay. So you have a beaker separating two different solution, kan? So side A and also side B. Solution A, solution B. So solution B is more concentrated. It has less free water molecule. Solution A, it is less concentrated. It has more free water molecule. So water will move from A to B, kan? From higher free water concentration to lower free water concentration, or from lower solute concentration to higher solute concentration. Okay. So,
Okay.
all right. Ada soalan? Any questions? That is osmosis. Okay. So although equilibrium is reached, so there's still movement of water, but at the same rate. Okay, at the same rate. Yeah, Afiq. Question from you? Tak ada?
Tak ada, salah tekan, salah tekan.
Okay. Okay, so now we are going to look at these terms. So that this term you are very familiar, which is iso, hyper, and hypotonic. Okay, so the main term that you have to understand is tonicity. Tonicity. So what is tonicity? Tonicity is the ability of a solution surrounding a cell to cause that cell to gain or lose water. Okay, so usually for any living cells, can living a cell, especially eukaryotes, single unicellular eukaryotes, or prokaryotes even, or even um, multicellular eukaryotes, can. So these are made up of cells. They are surrounded by solutions. They are surrounded by water. All living things requires water, right? Okay, so now you are going to understand that the different types of apa solution surrounding the surrounding that cell. Okay, so the solution can be hypertonic, it can be hypotonic, it can be isotonic. It depends on the type of cell. Okay, so if it's a plant cell requires a lot of water. So this water requires by the plant cell for the process of photosynthesis, for example. So and also to cause the plant to be turgid, right? So this water will be stored in the central vacuole and causes the plant to be turgid. So for plant cell, the normal solution that has to sorry, the normal solution that surrounds the plant cell, it has to be hypotonic. Okay. For animal cells, can. So animal cell, because we doesn't have any central vacuole and then there's no cell wall. So for animal cell to be able to survive, they have to be in isotonic surrounding solution. Okay, kita pause kejap ya. Okay, so tonicity. Ability is the ability of a solution surrounding a cell to cause that cell to gain or lose water. Okay, so this is the definition that is from Campbell, a reference book by Campbell. Okay, so we have three different types of solution: isotonic solution, hypertonic solution, and also hypotonic solution. So isotonic solution, its solute concentration is the same as that inside of the cell. Okay, so there will be no net movement of water across membrane. No net movement means that there's still movement of water in and out of the cell, but at equal rate. So that one you have to understand. Okay, so isotonic solution. So its solute concentration is the same, right? And then hypertonic solution apa? Equal rate. Okay, so equal rate, the movement of water in and out of the cell is at equal rate. Okay, so definition, the terms is no net movement of water. Okay, next solution here is hypertonic solution. So its solute concentration outside is greater or higher than inside of the cell. Right? So this causes water from inside of the cell to diffuse out through the process of osmosis. Water moves out through the process of osmosis. Cell loses water via osmosis. Okay. Movement of water also through osmosis. Any movement of water across plasma membrane, we call it as osmosis. And then this one here, hypotonic solution. So where the the solute concentration of the solution is less or lower than inside of the cell. So because there's more free water molecule outside, can more free water molecule. So this causes water molecule from outside to diffuse into the cell through osmosis, through by the process of osmosis. This because this is the plant cell. So when cells is immersed inside hypotonic solution, the plant cell, so the plant cell will be turgid. Okay, turgid. So this one, the in hypertonic solution, the plant cell will be plasmolyzed. As you can see, the process of plasmolysis occurs when water diffuses out and the plasma membrane of the cell will be pulled away from the cell wall and the cytoplasm shrinks. Okay, so okay, so this one it gives you the condition of animal cell. In this case is the red blood cells when you immerse in the three different types of solution. So red blood cell will lyse if you immerse the blood cells in hypotonic solution. Okay, in hypotonic solution. Hypotonic solution, there is more free water molecule causing water to diffuse into the cell, get through the process of osmosis, and causing the cell to swell first, and then burst or lyse. So you can use both burst or lyse. Okay, water enters because animal cell doesn't have cell wall, so this causes the cell to be burst as more water enters. Okay, for the second diagram, if you immerse the cell in isotonic solution, okay, so in isotonic solution, because the concentration of solute inside and outside of the cell is the same, so there will be no net movement of water. No net movement means there's still movement of water, but at the same rate. Water moves in and then move out, moves in and then moves out. Okay, diffuse in, diffuse out. Okay, the movement of water occurs at the same rate, in diffuse in and also diffuse out. So water flows across the membrane at the same rate in both direction. That is the definition here. No net, water moves at the same rate in both direction. So the volume of an animal cell is stable, which is isotonic, same. So for animal cell, for animal cell or eukaryotic cells, they have to be in isotonic solution. Okay, the normal condition. And then if you immerse the animal cell or the blood cell in this case in hypertonic solution, okay, so the solution outside is more concentrated. There's less free water molecule, and inside cell there is more water. So water from inside cell diffuse out. Can so cell will lose water to the solution. Okay, to its environment, the solution surrounding the cell. So because water moves out, diffuse out through the process of osmosis, it causes the cell to shrivel, and then it can lead to the death of the cell. Okay, so example of increasing salinity of lake to animal. Okay, so effects of increasing salinity of a lake to animal. So only certain organisms that can live in water that is highly concentrated. So they have to have some mechanism, such as, for example, um, unicellular eukaryotes that lives in really saline solution. They can have some form of osmoregulation to regulate the concentration of the cell. Okay, so fish ke kan, so marine fish kan, so marine fish, so they have some kind of mechanism to regulate osmo regulation. Okay, mesti kena ada mechanism to regulate the excess loss of water from the body. Okay, okay, kalau tak silap saya, contohnya kalau macam marine fish, they will secrete really small volume of urine that is really concentrated. So that is one way of osmoregulate. Okay. Okay, so next, so this one, if you immerse plant cell in the three different types of solution. So the first one, if you immerse plant in hypotonic solution, it causes the plant cell to be turgid, but the plant cell will not burst because it has cell wall. Kan? So water enters the cell by osmosis. When you're describing the process, you must mention the word osmosis. Water enters the cell by osmosis. Okay. Kita break sekejap. Okay, so let's continue. Right. Okay, so so plant cell, when it immerses in hypotonic solution, it causes water to enter by the process of osmosis, but the plant cell will not burst because it has cell wall, causing the plant to be turgid, very firm. Okay, so this is very important for plants such as herbaceous plant. Okay, herbaceous plant. And then the second diagram here, as you can see, if you immerse plant cell in isotonic solution. Okay, so if you immerse plant in isotonic solution, the solid concentration is the same inside and outside. So there will be no net movement of water, and then no change in cells volume. The cells become flaccid and may lead to the wilting of that plant. Kan? So dia tak jadi segar lah, dia tak jadi segar. Plant ni mesti dalam keadaan segar, turgid, right? Flaccid. In isotonic, the plant will be flaccid, and then the plant cell will be flaccid. And then if you immerse the plant cell in hypertonic solution, so it can lead to the plant being plasmolysed. Can so there is a net movement of outflow of water by osmosis from inside to the outside of the cell. So the cell vacuole shrinks, okay? The plasma membrane pulls away from the cell wall and the cell shrivels. So this is the phenomena, we call it as plasmolysis, can causes the cell to wilt even more, okay? So and then so this is for plant cell if you immerse in three different types of solutions. Okay, so the third type of passive transport after osmosis is we are going to look at on facilitated diffusion. Okay, so it involves the movement of solute across a membrane with the help of transport protein. Transport protein, which is transmembrane protein that are embedded in the plasma membrane. Follow concentration gradient means that from high concentration region to low concentration region, from high solute concentration to low solute concentration. So, increase the rate of diffusion across cell membrane. Uh, by having transport protein, this will increase the rate of movement of solute across the plasma membrane from outside to into the cell or vice versa lah. Okay. So each transport protein are very specific. So it means that in order for you to move water molecule across plasma membrane using transport protein, it requires transport protein. We call it as aquaporins. You cannot transport sugar through the aquaporins. Okay, so that is what it is meant by very specific. Specific transport protein transfer specific solutes. Okay. So and then there's two types of transport protein, channel proteins and also carrier proteins. Okay. So this is channel proteins. Kan? So as you can see, it has channels. Kan? So the type of molecule that can pass through channel proteins such as water, okay, and also ions. This one here is carrier proteins. So carrier protein can transport ions and also polar molecules. Can so carrier protein, as you can see, it has binding site for the solute to bind. So this is binding site of the carrier protein. So the function of the binding site is for solute to bind. As solute binds, it causes the carrier protein to change shapes and transfer the solute pass the membrane. Okay, so because it is passive transport, as you can see, the movement of solute from high concentration region to low concentration region, from high to low. Okay. Okay. uh, Okay, so for channel proteins, so channel protein provides passageways that allow specific molecules or ion to cross the membrane. Specific molecules or ions. Okay, so have hydrophilic passageways that allows water molecule or small ions to flow very quickly from one side of the membrane to another. Okay, so they have hydrophilic passageway channels that allows ions. Can ions have the channel because ions will have charge. They have charge, either negative or positive charge. So ions, although the size is really small, because they have charge, they cannot just diffuse through the phospholipid bilayer. They have to use channel proteins. Okay. Same goes to water molecule. Water molecule is special because water molecule can diffuse across the phospholipid bilayer. Dia boleh lalu across the phospholipid bilayer. But the movement of water molecule crossing the phospholipid bilayer is really slow. Why? Because water is a polar molecule. Air boleh tembus phospholipid bilayer tapi pada kadar yang sangat slow rate, kan? So slow rate. To increase the movement of water molecule across the plasma membrane, water moves along across the membrane using aquaporin. So this increases the rate of diffusion of water across the membrane if the movement of water is through aquaporin. So example channel protein are aquaporins and also ion channels. Okay, so explanation on aquaporins. So aquaporins are water channel proteins, facilitate the massive movement of diffusion of water molecule in plant and also animal cell. facilitate the massive amount of diffusion of water. So in order for water molecule to increase the rate of diffusion, it has to use aquaporin. Okay. So without aquaporin, the rate of water movement across the phospholipid bilayer is low. Water can cross the phospholipid bilayer but at slower rate. So to increase the movement is through the channel protein, is through the aquaporins. Okay. So the movement of water molecule across the membrane using aquaporin is also considered as osmosis. Kan? Asalkan dia melibatkan pergerakan air, kan? Pergerakan air that crosses the phospholipid bilayer with or without channel protein. So the process is osmosis. Okay. Okay. So ion channels. Okay, tadi aquaporins, now is the ion channel. So there's several types of ion channels. So the first one is non-gated or leak ion channel. Okay, so ion channel, non-gated or leak ion channel. So from the word leak or non-gated, so it means that the channel is always opens. Kan? So always open and responsible for the permeability of specific types of ion. Okay, specific types of ion simply allows ions to pass through the channel without any impedance. Okay, without any uh, halangan lah kat situ. So as you can see here, you have the leak ion channel for sodium ion. You also have leak ion channel for potassium ion. So they are specific. Leak ion channel for sodium, leak ion channel for potassium. Potassium cannot use this one. Sodium cannot use this one. So they are very specific. Okay, they are always open. Always open means that it allows the ion to diffuse down concentration gradient. Okay, diffuse down concentration gradient. Further explanation on leak ion channel is application where you are going to come across this term next semester in the chapter of nervous system. Okay. Okay, so next we have gated ion channels. Kan? So gated tadi non-gated. Kan? Non-gated, it is always open. The second one is gated. Ini second ya. The second one is gated ion channel can open or close in response to a stimulus. So the stimulus can be chemical ligand, chemical molecule. Chemical molecule we call it as ligand. Okay. Electrical or change in voltage or mechanical. So gated lah. Okay. So gated, for example, okay, gated kita ada voltage gated ion channel. Kita ada lagi satu ligand gated ion channel. Okay, ligand gated ion channel. Example of ligand gated ion channel is ligand is chemical molecule kan. So example of chemical molecule we can have is neurotransmitter. Okay, so in order for the gated channel to open, for example open, it has to have some kind of stimulus. So if you kalau digambar kat sini, okay, ni ligand gated channel kan. So ligand gated channel open or close in response to ligand or chemical molecule binding to receptor such as neurotransmitter. Example of neurotransmitter is acetylcholine. Okay, if you look at the first diagram here, this is example of ligand gated channel. Okay, for ligand gated channel, it has binding site. It has binding site. So this binding site is for ligand molecule to bind. Okay, ligand, chemical molecule to bind. Example of chemical molecule or ligand is neurotransmitter. Ligand is chemical molecule such as neurotransmitter, such as acetylcholine. Okay, so initially when there is no neurotransmitter binding to the ligand gated, the channel will be closed. Kan? So if it's closed, it will not allow ion to pass through. So what causes the channel to be able to open? You have to have the neurotransmitter binding to the binding site. So when neurotransmitter binds to the binding site, it causes the channel, the channel protein to open. So once channel protein opens, it allows ion to pass through. In this case, is sodium ions. Okay. One of the place where you can find ligand gated channel is at dendrite of neurons. Okay, dendrite of neurons. Can so you have, for example, one neuron and then another neuron. Can so one neuron is transmitting neuron to another. Sorry, it's transmitting impulse to the next neuron. In order for the neuron to transmit impulse is actually is due to the movement of ions from into the cell and out of the cell, causing voltage of membrane to change. Kan? Membrane to change. Sel, saya dah explain hari tu kan. Sel dalam keadaan normal dia, inside cell it is negatively charged. Okay, negatively charged. Outside is positively charged. So outside there will be a lot of sodium ion. Kan? So a lot of sodium ion. So once the channel opens, kan, it allows sodium ion to enter. Once sodium ion enters, it changes the voltage of the membrane. Yeah, so it changes the voltage of the membrane and allows the next neuron to be able to generate electrical impulse. Okay, that is where you can find ligand gated at dendrites of a neuron. Okay, a dendrite of neurons. Okay, so next is voltage gated channel. Okay, voltage gated channel. What causes the channel to open is it is because due to the change in voltage of the membrane. Okay, so the change of voltage. Initially, I said before, cell will be negatively charged. For example, if I'm not mistaken, cell tu dalam keadaan resting negative 70 millivolt voltage dia dalam keadaan rest, not receiving any transmission of impulse. But when sodium starts to enters, kan, the membrane will become slowly we call it as depolarized, become more positive kan as sodium ion enters. So dia akan jadi daripada negative 70 dia akan jadi slowly kepada negative 55 and then lastly dia akan jadi positive 30 millivolt. Kan? So this is the peak of an action potential. Alright. So due to sodium ion enters, more sodium ion enters and inside becomes positively charged and jadi voltage dia jadi positive 30 millivolt. Okay, so the change in voltage, for example, from negative 70 to positive 30, okay, it causes the voltage gated channel to open and allows more diffusion of sodium ion, for example. Okay. So voltage gated ion channel, open or close in response to small voltage changes. For example, voltage gated sodium channel, which can be found along axon of a neuron. Okay, axon of a neuron. You have to recap back the structure of neuron. You have the cell body. Daripada cell body will be dendrites, right? Dendrites is where you are going to find ligand gated. So dendrites, cell body, and then from the cell body it extends into axon. Axon is where impulse will be generated. Okay. So in order for the axon to generate impulse, along the axon there will be voltage gated ion channel. Ada dua jenis, one for sodium, one for potassium. Okay, so voltage gated channels will open or close in response to changes in voltage across membrane. Okay. So and then we have the third one is mechanically gated channel. Okay, open in response to physical deformation of the receptor. Yeah, so due to pressure, such as pressure, for example, as in sensory receptor of touch and also pressure. Again, so such as in the skin or ear, for example. So this is where you are going to find mechanically gated channel, right? Open or close in response to touch or pressure, right? Yeah.
Saya nak tanya, kan ada cakap sodium masuk dan nanti dia tukar charge kan. Dia panggil term dia apa? Neutralize ke?
Saya tak dengar lah apa awak cakap.
Bisa nak tak tanya? Sama.
Dia cakap kat ujung, awak guna phone ke?
Ya. Dengar tak? Okey. Tadi Madam cakap sodium masuk kan. Lepas tu nanti yang kat dalam tu daripada negatif dia jadi positif. Kita panggil term tu term apa? Dia neutralize ke?
Itu kita akan tengok semester depan. Dia ada
Okay.
term dia. Depolarize. Depolarize.
Alright.
Depolarization daripada negatif jadi positif. Okay, depolarization. Daripada negatif jadi positif is depolarization. Disebabkan sodium masuk. Nak jadi negatif balik, dia bukan sodium keluar. Dia ada ion lain yang keluar which is potassium. Detail of that process is next semester. Okay, so for now you just have to know, okay, ligand gated in response to neurotransmitter binding to it, causing the channel to open. Voltage gated is open or close in response to change of voltage across membrane. Okay. Mechanically gated, open or close in response to pressure. Okay. Yang positive, negative, 30, 70 ni you don't have to understand. I just give you an example of the condition. What causes the voltage gated channel to open, macam tu, or close. Okay, so this is just example where you don't have to know for now the change of voltage tu. Okay. Okay, so next is we have here disease related to channel protein. The first one is cystic fibrosis. Okay, cystic fibrosis is a genetic disease due to a mutated genes. Okay, so causing channel protein for chloride to be defective or absent. So important to regulate the flow of water and salt across membrane of epithelial cells. So buildup of mucus in trachea, lungs, pancreas, liver, gallbladder, reproductive organs, sweat glands and other organs. Okay, basically cystic fibrosis is a genetic disease. It means that it is due to mutation in your genes. Kan? So we know that your nucleus contains DNA that codes for many genes to synthesize specific protein. So in this case, the gene in your DNA is mutated, and the gene that is mutated involves in the production of channel protein for the transportation of chloride ion. Okay, so kita ada DNA yang besar, kita ada banyak gene dalam DNA. Untuk orang yang ada cystic fibrosis, the gene that involves in the synthesis of channel protein is mutated. So mutated, causing the synthesis of the channel protein to not occur, absent, tak boleh nak hasilkan channel protein tu at all, ataupun channel protein boleh terhasil but cannot function. Because the gene that code for the synthesis of the channel protein is defective, mutated. Okay, so it leads to the synthesis of defective channel protein or doesn't produce at all. Okay. So what is the importance of this channel protein? It is to transport chloride ion, okay, in and out of the cell. Okay, important to regulate the flow of water and salt across membrane of epithelial cells. Due to the channel protein being absent or cannot function, it causes mucus to build up in these organs. The trachea, the lungs, the pancreas, the liver, the gallbladder, the reproductive organs, sweat glands, and also other organs. Okay, so this is the explanation for cystic fibrosis. For example, in the lungs. Okay, so in the lungs, the chloride channel transport chloride ion from inside the cell to the outside of the cell. Okay, so we have the cell, we have the chloride ion channel. So it is to transport chloride ion from inside to the outside of the cell. Chloride ion move outside. Okay. So extracellular chloride ion is required. Okay, so you have to have chloride ion outside. Extracellular chloride ion is required to maintain healthy mucus as it attracts water. Okay, so when there's more chloride ions, so the presence of chloride ion will attract water, so causing the mucus production not to be so thick. Okay, so high concentration of chloride ion inside cells also accumulate sodium ions. Okay, so high chloride ion also attracts sodium ions. Okay, so causing sodium ion to accumulate. So causes mucus outside cells to become thicker and stickier than normal. So thick mucus cannot be swept away by the cilia, leads to persistent lung infection, chronic bronchitis, and pneumonia. Let's look at the next slide here. Okay. So this is the flow of how the channel is made. Okay. So CFTR. CFTR stands for the chloride ion, the name of that chloride ion. Cystic fibrosis, transmembrane, transmembrane protein. Okay, transmembrane protein conductance regulator. So that is the name. Cystic fibrosis transmembrane conductance regulator. The name of the chloride ion channel tu tadi kan. The name of this channel is CFTR. Okay. So in your DNA, there will be the gene that code for the synthesis of the protein. Yeah. So this is the protein synthesized. Okay. So once complete, the CFTR protein moves through the cell to the cell surface. So this protein is called as trafficking. So to allow movement of ions. So once at the cell surface, the CFTR protein function as chloride channel. Can function as chloride channel. So this channel helps maintain the right balance of weight in the airway, in the lungs, for example. Okay. So for normal CFTR, moves chloride ion to the outside of the cell. Pada keadaan yang normal, orang yang sihat, the chloride ion channel, we call it as CFTR protein, allow chloride ion from inside to move outside. Okay, from inside to move outside of the cell. So for the gene that is mutated, again, so it causes the channel to be defective or absent. Okay, so chloride ion from inside the cell cannot move out. Kan? So mutant CFTR channel does not move chloride ion, causing sticky mucus to build up outside of the cell. Okay, so when chloride ion cannot move out, kan, so it causes mucus to thicken up, kan, as water cannot be attracted to that area. Kan, tadi perlukan chloride ion kat luar, and then also attract sodium ions to also attract water, causing mucus not to be so thick. But when chloride ion cannot move out, the mucus become thicker. Okay, become very, very thick. So that is cystic fibrosis. So it occurs in this area. Kan? So the trachea, the lungs, pancreas, liver, gallbladder, reproductive organs, sweat gland, and also other organ that capable of producing mucus. Okay. Ini disebabkan oleh ni. This is the mutated part. Mutant channel. Ataupun tadi CFTR channel tu tadi kan, mutant. So the channel mutated, causing chloride ion cannot move out and causes sodium ion to accumulate. Macam itulah kan. So it causes mucus outside to become thicker because the ion cannot attract water molecule. Boleh faham tak? Can you all understand? Boleh? Okay. That is cystic fibrosis. So I will highlight again. Cystic fibrosis is when there's a gene that codes for the synthesis of the channel to be mutated. So the channel cannot be synthesized, or when if it's synthesized, it cannot function. So when it cannot function, the channel cannot move chloride ion outside, and causes mucus to become very thick because there's no chloride ion, and that condition will not attract water molecule, cannot attract water molecule, causing mucus to thicken up. Okay. So this is just to show you the location where there's formation of the thick mucus in the lung. Okay, so these are the areas that where mucus can become thick. So you can scan this QR in your own time for you to watch more explanation on cystic fibrosis, just for your general knowledge, again. So you can scan the QR to watch more explanation on cystic fibrosis. Okay, so for now we are going to end our lecture. Do you have any questions regarding today's lecture? Ada?
No.
Tak ada So kalau tak ada, kita stop dulu kelas for now. So we are going to continue our lecture on Friday. So on Friday kita akan tengok lagi satu, ada jenis penyakit lain, okay, yang berkaitan dengan transport protein. Okay, so class, so thank you.
Thank you, madam.
Okay.
Thank you,
Thank you, madam.
madam.
Okay,
Thank you.
alright.